Uh, cool. All right. We'll get started here. Just a couple quick announcements. Saw that a good number of people have gone on, completed the pre-screener. That's good. Looks like we're up to about 70, which is fantastic, but still want to make sure that people are going through completing that initial online screener. Again, what this is going to do is it's going to get your first hour of research out of the way. And then two, it's going to make you eligible for more likely to be eligible for stuff down the road. So definitely get on. Make sure that you do that. Also got a lecture from from Wednesday uploaded. So there was two. There was a little piece to finish off brain structures, and then the one that we went in to go and take a look at sleep. Looked and sort of views on that are still pretty small. I guess that's a good way to. I'm assuming that the general world isn't interested in popping into my Psych 1000 lectures. So just do please make sure that you go through, watch those videos, take careful notes. Again, quiz 2 tends to be a beast in terms of people's performance. It's the first quiz that we've got. Everything, a full set of lecture materials. It's also bio stuff, which tends to give people fits sometimes. So just make sure that you're getting on there, watching that lecture, taking notes, so that you're prepared and ready to go. Okay. Any questions before we get started on today's material? Anything where we're going, what things are going to look like, anything we've covered up to this point? Nope. Okay, cool. Then we will get into perception and sensation stuff, which is pretty cool. So we're going to go through, run through some initial material today. This is covering modules 16, 17. What I'm also going to do is I'm going to send you out a link to, has anybody, give me a shout out, has anybody watched the show Brain Games? Or seen it? Okay, Hannah, you have. You're the only person who's popped up in my window right now, but cool stuff. Just looking at the psychology of perception, and sort of this is an area for cognitive science. And our working assumption is that the world is how I believe the world to be, and it's just kind of sort of what I see is what you get. What we actually see without too many difficulties, you can start to play with how your brain is interpreting certain types of information. Super cool. You don't have to watch it, but I would recommend it. It's just kind of cool to go through and see, and it'll add to some of the stuff that we're talking about today. Cool. So, just some fundamental concepts to go over real quickly. And if I'm a cognitive psychologist working in this area, this is kind of sort of some things that I'm teasing apart. Sensation, okay? If we're talking about sensation, this is specifically the process by which our sensory receptors and our nervous system receive and represent stimulus energy. Okay, you can think about this as being kind of the bare bones basics, kind of the mechanics of things. So right now I'm talking and you're hearing words coming in. So the sound of my voice is coming in. There are receptors in your auditory system that are picking this up, that's taking that physical waveform, translating it into an electrical signal that's then transferred through your nervous system, and then it's going through up into your brain, right? So this is all about sensation, how we go through and collect and grab information and sort of go through and bring it into our brain. Perception is the process of organizing and interpreting sensory information. So you're hearing things. The physical waveforms coming through the mic on your speaker, whatever you're listening to, is producing triggering receptors in your ear that's sending electric signal back into your brain. But that electric signal is just electricity. There's nothing meaningful about that. Perception involves how your brain goes through and takes that information and starts to organize it, interpret it into something that you experience as spoken language and something that you can understand. Okay, so the difference between sensation and perception there. If we're talking about transduction, this is a basic physics, sort of engineering concept. Same thing we're using in these areas of cognitive psychology. talking about the conversion of energy from one one form to another. So again, if we're talking about the your auditory system, as we're talking or as we hear noise, there's a physical mechanical waveform that's going through air, right? And so what our auditory system is doing is taking that physical waveform and translating it into electrical signal that our brain can process. This would be a process of transduction. Okay, we're just taking energy from a physical form, translating it into electrical form, and then sort of going through and doing stuff with it there. Okay, psychophysics. This is going to be the study of how physical characteristics of different stimuli are translated into corresponding psychological experience. So, let's see here. That, right? Someone tell me what color that is. Yellow, exactly. Okay. And so what we're doing with psychophysics is the study of what characteristics of this actual physical stimuli are being translated into my perception of yellow, right? Sort of what is yellow, what features, what sort of physical features of that object are being translated into something that's perceived by me as a yellow object. Okay, so that's psychophysics. Any questions about sort of how we're teasing apart sensation, perception, transduction, psychophysics, people sort of getting the picture of what some of this stuff is? Okay, other thing too, everybody, look at me real quick into the camera, right? Real quickly, what does your shirt feel like on your shoulders? Okay. Before I said that, usually if we're in class, I'll say, what do your shoes feel like on your feet? Because I'm assuming everybody wears shoes. I don't know if people are wearing shoes or socks or any other such thing. But when we're talking about sensory adaptation, before I mentioned how your shirt feels on your shoulders, you probably weren't noticing that. Okay. And that's because what your body does is it has, through processes of sensory adaptation, if we start to have a stimuli that is just kind of persistent and it's not relevant, your body will start to sort of process that out. It'll start to adapt to that stimuli so that you don't really notice it anymore. Okay. So you can feel the sensation of your shirt sitting on your shoulders right now. Okay. But it's not relevant information for what you're doing. And so your body kind of gates it out. Now, if I draw attention to it, I can say, okay, yeah, I know what this feels like. But what this does is your body going through and starting to gate out things that if we were bombarded and aware of it all the time, it would start to become overwhelming, right? And so...
Is that the same thing as like when you become aware of your breathing and you have to start doing it manually?
Kind of, a little bit. Maybe the sensation of breathing, right? Like, so we're breathing every day. Now, if I go through and I have you focus on your breathing and the sensation of your chest rising, falling, things along those lines, I'm drawing attention to a physical sensation that you wouldn't otherwise be noticing. Now, breathing itself, that's just an automatic process that happens, right? Our body just does it. Like, we can stop it for a while, but I can't hold my breath until I die. My body won't let me do that, right? That's more of an automatic type of thing. If we're talking about adaptation, here's another example, all right? No one has to give a shout out, but let's say if anybody has a roommate, right? In COVID times, who knows who has roommates or not? But sometimes we have roommates. Sometimes our roommates are smelly, right? And you might walk into your room from the outside, you're like, it reeks in here. You are a disgusting human, right? But once I'm in that room for a while, do I continue to notice that? Nope. My brain gates it out. My brain's giving me a lifeline and saying, all right, it's smelly in here, but I'm going to go through and filter this out so that we're not constantly noticing it, right? So again, this is just this process of adaptation. All right. So just some fundamental concepts. But if I'm a cognitive psychologist, some of the types of things I start to become interested in, if I'm sort of in this area of perception, starting to worry about and think about, like, what are the absolute thresholds, or sort of what are the minimal levels of things that I can sort of hear, adapt, and sort of process as human beings. And so one of the concepts that we get into is this idea of an absolute threshold. Okay, absolute threshold refers to the minimal stimulation required to detect something at least half of the time. Okay, so think back. If you were in elementary school, anybody got called in to do hearing tests in elementary school? They still do that? Okay, yeah, okay. So again, in this situation, right? Like you put the earphones on, and who gave me a Michael? Was that you that said you remember hearing tests?
Yeah, I had to take them pretty much all throughout, just for different things.
Okay, and so what they did, right, Michael? They put headphones on, right, and then they start playing tones, right. And what you have to do when you hear a tone, you raise your hand, right, to say, "Oh, I've heard it in this ear, that ear, whatever," right. We can think about this as would be a nice way to go through and try and find the absolute threshold for auditory stimulation. Like how subtle, how small, how slight of a sound would I need to be presented with so that I could detect that at least 50% of the time. Okay. And this 50% of the time piece is important because I might catch something one time and not catch it the other time, right? So what we do with these studies is we would go through and let's say we present something at, I don't know, like 5 decibels, repeat it over and over and over again. And if you can't detect that 5 decibel noise at least half the time, then that would be below your absolute threshold. But let's say we kick it up and it's like, say, 12 decibels or something like that. And Michael's able to, at least the half the trials that I present him with a 5 decibel noise, he's able to identify that. We would say that that was the absolute threshold. And so what we do, because different people have different levels of hearing, large group level studies, we can start to detect the absolute threshold for certain types of stimuli. Okay. So we're going to do a demonstration here. And it'll work a lot better here probably than it does ever in the classroom. So points for COVID, I guess. What I want you to do is on your screen, focus right between those two arrows there. Okay. Everybody looking? Did anybody see what that word was that flashed on the screen? All right, let's try it again. Anybody catch that one? Utah? No, wasn't Utah, but it might have been. It wasn't, but it could have been. All right, let's try it again. Anybody catch that one? All right. One more. Let's try this. Anybody see that? Okay, sweet. Because what happens when we're in actual class, the delay between the projector and the computers like slows everything down, and so like everybody's able to identify the first one right away. So this is actually a better thing. But what I'm doing here is I'm going through and I'm throwing on with the very, very precise capabilities of PowerPoint, displaying certain words at very, very low level or very, very quickly. And so technically this should be below the threshold for visual identification. Okay. These words are being flashed on there too quickly for you to go through and say, okay, that's what this thing is. You see something came up, right? But it's below the threshold of sort of being able to identify it. As we go through each one of these examples, the target word is put on a little bit longer each time to where by the time we get to this last one, you could probably notice it if you were me looking at the screen. But so people get the idea of kind of what we're doing with this demonstration. Okay. All right. So if we're thinking about absolute threshold, right? This is again the least amount of stimuli that we're able to reliably detect at least 50% of the time. But people who are working in cognitive psychologists working with signal detection theory. Okay, here I got people coming up on the chat. All right. Yeah, Jacob, the last one was happy. Nice work. Very acute sense of vision there, right? Okay. Signal detection theory. So if we go through and we figure out the absolute threshold, an absolute threshold is something that we're studying within the confines of a sort of a lab, right, with no distractions, everybody's paying attention and things like that. Signal detection theory, this is going to be factors that start to influence the absolute threshold of some stimulus at any given time. So, Lindsay, you're right here in my screen right now, okay? If I was having you go through and determine the absolute threshold for some type of sound or some type of thing like that, you might have a different absolute threshold if you were very, very tired versus if you were alert and awake, right? Or if, let's say, I did something to make you feel emotionally scared or angry versus content and calm. What that might do is that change in that psychological state might start to go through and change the absolute threshold for being able to detect certain types of things. Okay, and this is sort of the idea behind signal detection theory, looking at, okay, this is the absolute best threshold under optimal situations or optimal circumstances. What happens if we start to change the physical, psychological state of an organism to see how that changes? Do we become more able to detect things at a lower threshold, or does it increase the absolute threshold so we need more stimulus to be able to reliably detect? Okay. This also gets into those stimuli that are presented below the threshold of conscious awareness. So for vision, right, with those stimuli that I was popping up, it's about 100 milliseconds before you can start to reliably detect something, right? Anything that I present to you below that threshold, what we would call a subliminal stimuli, right? And what's really, really interesting is we have evidence that your brain is processing it, right? That information is getting into your brain. It's just not at a level that you're able to identify, verbalize, and things along those lines. Now, presentation of subliminal stimuli it's very subtle, and the effects are very weak. It's not brain mind control that you see in TV and movies about like, oh, here's the subliminal stuff going on in this. If you play the record backwards, it's going to tell you to worship the devil type of thing. But we do have evidence that it has an influence. And what we're going to do when you get into social psychology, talking about racism, prejudice, sort of things like that, we see that in laboratory settings, we can use some of these stimuli to start to prime certain sort of schemas and things like that that actually can go through and influence behavior. Okay. So, oops, here we go. Get that back on this. So this is absolute threshold, okay? We also have within sort of this area talking about difference threshold. Difference threshold, absolute threshold, what's the minimum information or the minimum amount of stimulation where I actually need to be able to detect that something's there or sort of perceive something. Difference threshold is the minimal difference, the least amount of difference between two stimuli to be able to detect that difference 50% of the time, okay? So if we go back to Michael's hearing test, right? Michael, this time, instead of having to identify, is that a sound or is there not a sound? What I'm going to do is I'm going to give you two sounds, right? And I'm going to say, tell me if these two sounds are the same volume or different volume, okay? Different threshold would be how much more intense does that second stimuli need to be in order for you to reliably detect that one was louder than the other at least 50% of the time, okay? So I'm going to play you two tones. Some of them are going to be exactly the same volume. Sometimes one is going to be louder than the other. How much louder does that second stimuli need to be in order for you to sort of detect the difference, okay? And what we find when we're doing this stuff, and it's super interesting, not many things in psychology conform to some type of consistent law, right? Something that is kind of a universal. But what we find with a difference threshold is very interesting, is that what we see is that two stimuli have to differ by a certain constant proportion to be perceived as different. So what this means is that the amount of difference, the absolute or the difference threshold for any stimulus is directly proportional to how intense that stimulus is to start with, okay? So again, let me see. I'm going to go out of my, who else can I pick on here? All right, Jacob, you like concerts?
Yeah.
You like concerts. Okay. So Jacob, you're at a concert. So if I'm right here, all right, you're listening to me right now and I'm talking like this, which one's louder? If I'm talking like this or if I'm talking like this, which one's louder?
Number two.
Number two. Okay. The way the level at which I raised my voice there is discernible because my initial thing was kind of sort of talking sort of minimally, right? But if you're at a concert, right, and you're in the front row and you've got sitting right by that amp and you're just getting that, how much louder those amps have to kick up in order for you to say, oh yeah, things just got louder? A lot, right? That's got to increase by a whole lot before you're able to, if you increase it by the same amount that I did right there, in that sort of when I'm talking at you, if you increase the amp by that amount of decibels, you're not going to be able to tell the difference, right? And so what we see is the amount of difference that we need is directly proportional to how intense the initial stimulus is, okay? So another example, thinking about mass, all right, so weight. What we see is that for mass, we need to increase the level of the stimuli by about 2% in order for you to perceive a difference, okay? So if we go through and we think about, does anybody have their textbook sitting next to them?
Yeah.
Who's got their textbook there? Jonathan? Okay. I got Jonathan in my screen right here. So Jonathan, increase, here I'm going to do this. Can you hold your textbook up there real quick for me? Okay. This textbook that Jonathan has, usually we would be doing this to have someone come down in front of the class and sort of do whatever, but we're not. So Jonathan, you're my demo person right there. So Jonathan's got that textbook. That textbook, your textbook weighs about 5 pounds, okay? And so what the difference threshold would say is what we would need, Jonathan, if I had that, you had that textbook in one hand and you had another textbook in the other hand, how much would I need to add to the other textbook? It's the... You tell the difference there. How much weight would I have to add to that 5 pound text in order for you to say, reliably say, okay, this one's more different than this one? And what we say is about 2% of 5 pounds is about 1.6 ounces, which ends up being, so this class is called What's on Clap's Desk. We got AAA batteries, right? I would need to add about the weight of 4 AAA batteries to that text for you to reliably detect the difference between this one's heavier than this one, okay? So again, because that 1.6 ounces, that's about 2% of that textbook, okay? Now, what about anybody in the class, lift weights, weight train, anything like that? Brett, did you raise your hand there? Okay, so Brett, let's say you're on a bench press and you have a plate on each side, okay? 135 pounds. If I added 4 AAA batteries to that, would you be able to tell the difference? No, you're not, right? What we would actually need to add a little over 2.5 pounds in order for you to reliably detect on those presses which one was different than the other one, right? 2.5 pounds is a lot different than this, right? But we're starting out with a more intense stimuli. So when I was talking and then shouting at Jacob, you detect the stimuli. I didn't have to raise my voice too much in order for him to tell a difference. But if he's standing in front of a concert, we have to increase that by a whole lot more before you're able to go through and tease that apart, okay? So that's going to be kind of the nuts and bolts of Weber's law. Do people kind of understand sort of how this works out? And it's pretty cool because it does really follow. It's different for different stimuli, right? Sort of the proportion we would have to increase sound or light or things along those lines differs, but it does look like it works in a pretty lawful, orderly fashion. So get back up here. So quick question: anybody notice any difference in the headings, the primary headings on this slide? Here. See, that's because I'm so sneaky, right? These are actually different size text. This is 20, this is 19.5, this is 20.5, right? So again, these are stimuli of different sizes, right? if anybody's looking at that, can anybody tell just looking at the screen that those are different sizes?
Very barely.
Very barely. So Cade's saying that he's able to detect a difference, right? Now, whether or not Cade could detect that difference at least half the time, that would start to get at sort of this idea of a difference threshold, right? And probably sort of getting into signal detection theory, unless I brought that to your attention, it probably would fly under your radar, right? But what I can start to do is I can start to play around, change your psychological state, your level of attention and things like that, and that starts to play around with some of these different thresholds. Okay. All right. So another big thing that starts to get into perception, sort of in terms of a principle, we talk about bottom-up processing and top-down processing, okay? If we're talking about, and this is a concept that's going to go into a lot of different areas. This is going to come up again and again. If we're talking about bottom-up processing, okay, what we're talking about, at least in sort of the field of perception, is things that are driven by just the basic physical characteristics of some type of external stimuli. You talk about this as data-driven processing. So let me see here. Who else? Brooke. What color is this circle here? What about the outline? What color is that? What about this? What color is this? Is this vertical or horizontal? Yeah. Okay. What you're doing, what Brooke's doing right here is all bottom-up data-driven processing. Being able to, if we were sort of virtually, or you go through, you could trace the edge of each one of those shapes, right? You can sort of identify the circle as a circle, just given the basic characteristics. And this is bottom-up processing. This is just sort of identifying basic characteristics, okay? But again, what we know is that our perception of different types of things isn't only influenced by sort of the physical characteristics. And this is where we start to get what we would call top-down processing. Top-down processing starts to be impacted by higher order mental processes, kind of your psychological state, things along these lines, okay? And what I can do is I can start to play around. Brooke, will you hang on with me and be my guinea pig for a couple more demonstrations here? Awesome. All right. What I can start to do is I can start to fool around with your perception of things based on sort of how you're looking at stuff. So for example, actually this is, okay, we're going to do something. I didn't have this set up. All right, Microsoft Paint. All right, cool. Freaking sophisticated here. All right, so if I go through, oops, not. There we go, I want that. Okay, I'm drawing this with my mouse. Kind of crude. All right. Usually do this on a chalkboard, and it's generally a sort of better representation here. All right. All right, Brooke, what did I draw right here? A cube. Okay, so if I'm looking at that, I'm looking at that cube, what part of that cube looks to be closest to you in the screen?
Probably the most front where it's square, the first one that you drew.
So to this one right here, right? So this one is the one facing closest to you. Can anybody go through and switch that around? Yeah, Jacob, as you switch that around, which one is the closest one?
The one on the top right, if that makes sense.
Yeah, right up here, right?
Yeah.
Brooke, can you switch your set to see that one is the closest?
Yes.
Okay, all right. So again, what we can start to do, it's the same thing, right? From a top-down process, right? Or from a bottom-up process, I can go through and sort of trace out the outline of this cube, right? But what I can start to do is I can start to change my perceptual set so that the cube is either coming out or going up, sort of which faces sort of popping out more directly. And this would be an example of changing your perceptual set, right? So depending on how you are approaching that, it makes it look like a slightly different object depending on your set there. Okay. I can also start to play around with, okay, oh, here's another one. Here's another good example. Brooke, because you're helping me out here. What do you see here? What is this a picture of?
A lady.
A lady. Tell me more about this lady. What's this lady doing?
She's looking at something in the distance.
Okay, so this looks like to be a young lady looking off into the distance, right? Anybody else see something different?
I see an older woman as well.
Okay, Kate, where do you see this older woman? Tell me about that.
She's looking down instead.
Okay, looking down, right? People see. Give me a shout out if you look at this. You see young woman. Jacob, Cade. Okay. Okay. Old woman. When you first look at it, who sees old woman?
I do.
Okay. Okay. Can people go back through? Can you go through and sort of see old woman, young woman in the same thing? Okay. This is an example of changing your perceptual set. Bottom up, it's the same picture. It's not different, right? But as I start to go through and play around, I can see a very, very different image depending on how I'm approaching it, right? And so this would be an example of top-down processing. We also see some stuff with context effects. What do we see here? Brooke, you're on a roll. Tell me what you see.
A mouse.
I see a mouse too, right? What do we see here? What's that image on the end?
You kind of have a bunch of people, and then at the end you have a mouse.
Okay. Does anybody not see a mouse at the end of that set?
Yeah, they're wearing glasses.
Okay. Yeah. So if I go through and I give you two different sets, right? If I give you, so it's the same image at the end here, right? But if I go line drawing a face, line drawing a face, line drawing a face, line drawing a face, I see a face, right? I might see a mouse, but I'm much more likely to see a face, right? But if I go line drawing of animal, line drawing of animal, line drawing of animal, line drawing of animal, I get here, what am I more likely to see? That mouse, right? So Brooke, you saw mouse right here, which makes sense, absent context. And then you saw that mouse, and then it kind of sat in your head. So that's a mouse, right? And so if I go through, you're maybe not as susceptible to that context effect. But if I hadn't shown you that image to start with, you would say, oh, you'd be much more likely to say, oh, that's a person, right? And so what we can start to do, depending on what I present to you before I present the target stimuli, I can start to change around because I'm manipulating your what you're expecting, what your sort of your frame of reference, and can start to change things. Okay. We also see impacts of emotion and motivation and things like this. Okay. So if okay, Brooke, you're awesome. I'm going to leave you alone for a hot minute. Okay. I say people without their cameras on. You're fearful. You're cowards. You're not cowards. It gives me less people to pick on. Heidi, you're sitting there. You're sort of doing some crocheting, but I'm going to pick on you for a moment. Okay. If we were doing, and we do this sometimes, we have what's called a cold pressure task. And any athletes out there ever sprain an ankle, or you don't have to be an athlete. Maybe that's why you sprained your ankle. You're not an athlete like me. So you have to put it in an ice bucket. Is that comfortable? No, that's horrible, right? And so what we do, we can't sort of, again, ethically inflict too much pain in experimental settings, but we can do things that are uncomfortable, right? And so if I were to have you put your hand in a bucket of ice cold water, right? Your perception of how cold that water was or how painful that is might start to change depending on sort of where you were to start with, right? Now, if you're in a pretty good mood, you're pretty happy, you're upbeat and things like that, you might identify that as painful, but not terribly painful. But if I catch you if you're anxious and nervous or upset or angry and have you do that, what you might identify is start to identify that water as being colder than you would have or more painful than you would have if you were in a different psychological state. Okay. Same thing. Anybody at any point playing football or on a football team, anything like that? Okay. Ish. All right. Let's say you're midway through practice, you're going to have a break, and this is what we had to do. You had to carry someone down to the end of the field, and then they had to carry you back before you could have water, right? If I'm fresh, if I'm right out there, how far do I judge that 100 yards to be? Far but doable, right? If it's midway through practice and I'm exhausted and I'm in a physically taxed state, my perception of that distance is longer than it would be if I was in a different psychological state, right? And we've gone through and sort of tested this out, and so you can probably note it from if you're running, right? This distance seems much longer to get to the finish line if I'm tired versus if I'm fresh, right? And so we start to see that, again, example of emotion, motivation types of things starting to impact your physical perception or your sort of psychological perception of actual physical distances and things along these lines. Okay. People comfortable with top-down, bottom-up type of stuff? It's pretty cool stuff. And again, when I send out that link, it's just a first episode of this show called Brain Games. It's just super cool to see sort of pretty easily how we can start to trick stuff. All right. Yeah. Ramon. Yeah.
Okay, so about like the whole process and stuff. What about someone who's colorblind?
Hold on to that. We'll get to that in a moment. Okay. Yeah. Again, you guys, I need to be paying you money to just set up the next slide. I like it. You know what this means? This means I'm doing such a fantastic job. Your mind, I'm manipulating your mind. Your mind's going exactly in the direction that I want it to because you're sort of anticipating some of this stuff. So, not that Ramon is easily manipulated. He's not. He's an independent person who does what he wants when he wants. All right. So we're going to go through, talk real quickly about basic components of the eye, anatomy of the eye. This is probably stuff that you got in third grade. But just to go through, make sure that we're all on the same page with this. Down here, we've got your basic diagram of the eye. You got your cornea here, your outside covering pupil, hole in the middle, iris. This is a muscle that's sort of contracting, relaxing to let light in. Here's your lens that's focusing information, right? Or focusing light coming in. And when light comes in, goes through the lens, what it does is it focuses it back here on your retina, okay? And your retina, from a perception standpoint, is where all the action is happening, okay? If we take a look at your retina, sort of a surface on the back of your eye, and what's interesting is when light comes in, sort of in through the eye, right? We've got these rods and we've got these cones that are going through and processing information, okay? Your rods have many more rods back here in your eye. Someone tell me what rods are important for doing. What do your rods do?
Picking up what your cones can't. So like black, white, dark.
Yeah.
Black, dim areas.
Yeah. Oh, Michael got that. Okay, yeah, Michael's exactly right. What your rods are doing is your rods are sensitive to light. Your rods have poor acuity, right? Your rods are low def. Your rods don't sort of pick up fine detail very well, but your rods are very, very, very good at detecting light sources and things like that, okay? What you have, lots and lots of rods in there, but then you also have cones. What do cones do? They pick up color. Cones are sensitive to color, and cones are highly sensitive to detail, okay? And so what we see is we've got two different receptor cells. You've got your rods and your cones performing complementary functions. Your rods, these are sort of picking up sort of basic light and things like that. Cones, sensitive to detail and color. Cones most highly concentrated here in a little divot in your retina known as your fovea, okay? And when light comes in, most of this is getting focused onto your fovea here, this is where you have the greatest sensitive to detail in light, or detail in color. So as you're looking at the screen right now, right, you're probably hopefully looking directly at it, right down the center. What this is doing is this image, light being reflected back, is focusing right there on those foveas, so you're able to read and identify information and things like that. Okay, so cones most highly densely packed into your fovea. Your rods more densely packed on your periphery. Okay, and so what this means is that if I'm trying to go through, let's say we're in sort of prehistoric times or sort of pre-civilization, and Hannah's trying to send me a signal because she's on or maybe not pre-civilization, it's a medieval times. You're in a castle, long, long way, far away, miles and miles away, right? But sort of bad news is coming, you need to send a signal, right? So you've got light a fire at the top of a tower, right? Am I most likely to detect that fire, that light far off in the distance, very faint, if I'm looking directly at it, or if I'm looking sort of outside on my periphery? What do you think, Hannah?
Probably looking directly at it.
You would think that, right? Actually, the periphery. Remember that your rods are highly, highly sensitive to light, right? And so if I'm looking if I'm looking for some light off in the distance, I don't need to see sort of features and color and things like that. What I actually just need to do, I need to pick up the light. And so what we see is in our periphery, we're actually much more sensitive to sort of light and sort of stuff going on. Maybe more so than our center, because in a direct part of our vision, that's where our cones are concentrated. So we're catching fine detail and things like that. If I'm going to read my notes here, right, I'm not going to hold it up out there because I can see that there's something there, but my cones aren't processing that. If I want to do that, then I'll hold it up straight, right? But if I'm looking just for sensitive, sort of picking up light, we're actually a little bit better at seeing that stuff in the periphery if all we're trying to do is detect things. Okay. So the interesting thing when we're thinking about, so we got a good handle on rods and cones, but we would think that if I was thinking about things, I would be thinking that my rods and cones are kind of pointed out towards and sort of catching the stuff that's coming in, right? In actual fact, it's not. It's the opposite. If we go through and take a look at our diagram here, we see that light comes in, and these are our rods and our cones. Business end of the rods and the cones are all facing back towards our brain, okay? And so what we have is light coming in through the pupil, coming back, being concentrated on the retina, and that light comes in through the retina and actually passes by a bunch of stuff until it hits the back of the retina, and then this is where rods and cones are picking up that signal, okay? The business end of the rods and cones pointing backwards, the light activates these cells, which then go through and send messages to their axons down here to some bipolar cells, right? So these are just connectors back to these ganglion cells, and these ganglion cells then kind of come together, form your optic nerve, and transmit that back to your brain, okay? So again, just in basic structure, light coming in, hitting the back of our retina, which is then triggered by the cells. They come together, organize those electrical signals from firing back into your optic nerve.
Dr. Class,
Yeah.
is there distortion in vision from the light being altered once it hits the retina?
I'm not sure. That's going to be like a sort of a kind of a deep tracks cognitive question here. I mean, probably Court, if it is, we wouldn't know because that's how we always perceive light and things along those lines. So, but I think you're hitting on the right thing, right? Because it seems strange that light would become refracted, distorted as it sort of has to pass through this stuff. Now, my sense is this sort of membrane back here is incredibly, incredibly thin. So if I had to guess, that's probably why it doesn't fool around. It's not like 3 inches of retina it has to get through. We're talking about just a couple layers of cells here. So probably it doesn't impact it a whole lot. But I'm not 100% certain on that. It might play around with it in some ways, but I guess we've adapted to address that. And I guess maybe if It helps if these were pointed the opposite way. I don't know. Maybe it burns them out or something like that. I don't know. But does that kind of help? Okay, yeah. Just I would think your head's in the right spot, and this is why I sort of brought it up, because it's kind of interesting to think that it has to go through a layer before it's detected. But my guess is that it's just so incredibly thin, it doesn't actually impact the light that's being focused back. Cool. All right. So when we start to think about vision is incredibly complex, and it's kind of our primary as we're visual creatures, right? Sight. We're sight animals, right? And so the processing of information in our world coming back through our eyes starts to become pretty complicated. So what we've got here is that your retina from each one of your eyes is going to receive information the most of it from both your right and your left visual fields. Okay, so do this for me, right? Look at me and sort of put up sort of your right hand. Okay. The people I can see in the screen. So I want you, there we go. Everybody else, you're off the hook, I guess. But if you take your finger and move it out and sort of keep moving around, if you're looking straight forward, right, until you can just start to detect it. Okay, that's your right visual field. That's this area right here. Okay, if you do the same with your left hand, and if you're looking straight ahead and pull your finger in until just so you can start to identify it, that's going to be your left visual field, okay? But what happens, as we see, is so my right visual field is being processed by cells on the left half of each one of my retinas. My left visual field being processed by cells on my right half of the retina, okay? And so if we take a look at this, what we see is kind of this crossed pattern, right? Where we've got information in my on my far right periphery, right there where I can just barely bring it in. That's being processed uniquely by sensors on the left side of my retina of both eyes, right? Things in my left visual field, just so it's right to where I can see it, being uniquely processed by cells on the right side of my retina, okay? Now, Hannah, what about all this stuff in the center of my visual field? So if I'm looking straight at my computer, which side of the retina is impacting that information, or is processing that information? Can you tell from this diagram? Okay. So in the middle here, this is stuff that's being processed by both. Okay. So what we start to see is the bulk of information, right? So I'm looking at you. Are you looking at me? You're looking at the slide, right? So to your image on the screen, I'm looking directly at you, is being processed by cells on both retinas, right? But if I go off onto the end, out here on my right, this is uniquely processed by cells on the left side of my retina. Here in my left visual field, this is being uniquely processed by stuff on the right side of my retina. But everything in the center of my visual field, both retinas are processing that. And this starts to become, or both sides of the retina, or each side of both retinas processing that. And where this starts to become interesting is our visual field. So my cells on the left side of each retina are going back through and being processed in my left hemisphere. Stuff in my right, stuff that's going being picked up by the right side of my retina, that's going back into the right side here, right? But what this means is that, Ramon, if I'm talking about information based on this diagram, information in my right periphery, what hemisphere of my brain is processing stuff in my right hemisphere, or excuse me, in my right visual field? The left hemisphere. And Ramon, things going on in my left periphery, where is that being processed? My right hemisphere, right? So we see this contralateral functioning. And so for normal everyday stuff that I'm looking sort of straight down the middle, right? That's being processed by both hemispheres of my brain because it's being picked up by both sides of the retina. And so that information is going directly to my right and left hemisphere. But if I get stuff uniquely, all this stuff that I can just coming into my right visual field, my left hemisphere is the only piece of my brain that's getting that. Same thing with my left, only getting into my right. And so what we can start to do is start to play tricks on some individuals. Now, right here, what we see is this information coming through, and our right and our left hemisphere are connected by what's known as the corpus callosum. Corpus callosum is a bundle of nerves that allows my right hemisphere to talk to my left hemisphere. But sometimes, for individuals with severe seizure disorders, there are procedures where people have gone through and severed the corpus callosum. The idea is that if a seizure starts on this side, it doesn't spread to the other hemisphere to try and sort of reduce that, right? And you would think that separating your two hemispheres would cause a lot of problems. In fact, it actually doesn't. It actually doesn't do too much, right? For the most part, we're able to sort of go through and function fine, but there are certain situations where this starts to have certain types of effects. And when we start to get into experimental situations, we can start to see what some of those effects look like. Okay. So we'll stop there. What we'll do is we'll pick back up and finish this stuff up in our next lecture on Monday. Okay. We'll do that. And then we'll finish up with non-visual senses. Non-visual senses is pretty straightforward business. But any questions about stuff that we've covered up to this point? Feeling good about it? Okay. So what I will say is that remember that next Tuesday we're going to have next Wednesday will be quiz 2. All right. Modules that you need to modules that you need to read and take notes for are outlined in your syllabus. Okay, make sure that you go through and you read that, and that you go through and you take careful notes. If you have access to Launchpad, the learning curve activities are a really nice way to go through and see if you've got a handle on that material. Go through and quiz yourself. Okay, make sure that you've watched all the lectures, that you've gone through and you've taken notes. Take a look at those comprehension check and lecture summary slides. Make sure that you got a handle on that stuff. Okay, again. This next quiz is like historically, and it happens every single time. It's a pretty reliable effect. We see sort of scores drop on quiz 2. Again, it's more material. It's sort of the first kind of full thing. People get thrown off by bio stuff sometimes. So just make sure that you're going through, that you're reading, and you're prepared. Do this early. You'll be way better off if you're going through and processing this stuff slowly, as opposed to trying to crunch everything there at the sort of the night before. Okay. Samantha, are you anywhere out there in the audience?
Yes, I am right here.
Okay, there we go. See, I knew Samantha's always there. She's lurking in the background, not like a creep, but just sort of there, like a reliable pal back there. Are you having SI? Is there SI session tonight?
No, our SI session will be on Monday.
Yeah, it's like. It's COVID Friday, Monday, sort of Thursday afternoon. Who knows, right? Okay, sorry about that. Yes. So SI session, sorry about that. Monday, I have a review session Tuesday. Same thing as we've always done with that. Just make sure that you're there, prepared, and sort of have started to process this information. So you got a handle on that. And then quiz will be the same setup as it was before. You'll get on, you'll log on again. You're going to make sure you got to log on right at 11 during class time. Go through, do that, and that'll be all we work on next Wednesday. Okay. All right. Other thing too. Oh, yep.
I will second what Josh said. I took his class, and you guys should all read the book. That will help you a ton. I promise. So.
Yeah. And again, you're going to have 10 questions on the quiz that'll be just from the text. It won't be stuff that we've covered. But then the biggest thing that the text is going to do is it's just going to double down. It's going to reinforce the stuff that we're talking about in lecture. It's going to give you a different angle on it. It's going to give you some different examples you're going to be thinking about a little bit differently. Make sure that you've got that information, not just, oh, I can identify things that I have before, but you're understanding some of these processes will become important. Okay. Other thing too, some of you have reached out wanting to go through and take a look over your last quiz. Again, if you want to go through, see what tripped you up, get in touch with me, get in touch with Natalie. Samantha, I don't know if you can access that stuff. We might touch base later and see if you can get through and access with the SI account. But basically, get in touch with us. What we'll do is we'll walk through the quiz. You can go through and look at stuff that you got wrong, and what that can start to do is start to help diagnose maybe where you got turned around, or sort of how to go through and improve preparation for these. Say, oh, okay, it looks like you're sort of maybe struggling in this area, that area. So happy, want to go through those that stuff with you. You're your own people. It's a free country. I'm not going to strong arm you, force you all to look over your stuff. But if you want to, let us know and we'll get you set up. Okay? All right. Any questions before I turn you loose? Cool. All right. Enjoy your weekend. Stay safe. And we will catch you all on Monday. Let me know if you have any questions. All right. We'll see you, folks.